For most of recorded history, the story of how people first arrived in Canada seemed settled. Textbooks had it locked down. Museums displayed it with confidence. Scientists repeated it in lecture halls from Vancouver to Halifax. A group of prehistoric hunter-gatherers crossed a frozen land bridge from Siberia into Alaska somewhere between 15,000 and 20,000 years ago. They spread south and east. They became the ancestors of every indigenous nation on the continent. End of story. Except it was never the end of the story. Because in the last 2 decades, ancient DNA pulled from bones buried beneath Canadian soil has been quietly dismantling that entire narrative. Not chipping at the edges, not creating minor footnotes, dismantling it. The genetic data now sitting in university servers across North America, Australia, and Europe tells a story so different from the one we were taught that some of the world's leading geneticists have described it as one of the most stunning reversals in the history of human prehistory. What the DNA shows is this: the people who first walked into what is now Canada were not a single group. They were not uniform. They did not all come from the same place, follow the same route, or arrive at the same time. And some of the genetic signatures buried in their bones connect them to populations on the other side of the planet. Populations separated from Canada by an entire ocean in ways that science is still struggling to fully explain. This is the story that ancient DNA is telling us, and it changes everything about who Canadians, and especially Indigenous Canadians, actually are. To understand why these discoveries are so explosive, you first need to understand what science thought it knew. The dominant model, which took most of the 20th century to solidify, was called the Clovis first hypothesis. It was named after an ancient stone tool culture first discovered near Clovis, New Mexico in the 1930s. The Clovis people were believed to be the first humans in the Americas. Their distinctive fluted spear points were found across North America, and for decades no confirmed human site predated them. The theory went like this: a population of ancient humans in northeastern Siberia, genetically related to modern East Asians, gathered in a region now submerged under the Bering Sea. During the last ice age, sea levels were dramatically lower, and a vast landmass called Beringia connected Asia to North America. These people lived on Beringia for thousands of years, isolated from both the Asian populations behind them and the uninhabited Americas in front of them. Then, around 15 to 16,000 years ago, as the ice sheets began to retreat, an ice-free corridor opened down through what is now Western Canada, and they followed it south. Within a remarkably short time, by this model, their descendants had reached the southern tip of South America. They diversified. They became the hundreds of distinct indigenous nations that Europeans encountered when they arrived in 1492. And they did all of this without any meaningful contribution from any other founding population. It was a clean story. It was elegant. And the ancient DNA is showing us it was wrong. The first major crack appeared not in Canada, but in a cave in Brazil. In 1975, a paleoanthropologist discovered a skeleton at a site called Lapa Vermelha 4 in the state of Minas Gerais. The skeleton belonged to a young woman who had died 11,500 years ago, making her one of the oldest human remains ever found in the Americas. She was nicknamed Luzia, and when scientists examined her skull, they ran into something they could not explain. Luzia's skull did not look like the skulls of modern Indigenous Americans or East Asians. According to the morphological analysis, it looked closest to the skulls of Indigenous Australians. This was so unexpected that many scientists simply dismissed it. The sample size was too small, they said. Skull morphology is notoriously unreliable, they said. But the skulls kept appearing. Across Brazil, Mexico, and further north, paleoanthropologists were recovering ancient skeletal remains with that same unusual morphology. Long, narrow brain cases, projecting faces, features that looked far more Australasian than East Asian. These people were being called Paleo-Americans, and they seemed to represent a founding population in the Americas that predated or ran parallel to the people who gave rise to modern indigenous Americans. Still, without DNA, it was possible to wave all of this away. You needed the genetic code itself. Then, in 2015, a team of researchers at Harvard Medical School, led by geneticist David Reich, published a paper in the journal Nature that made headlines around the world. The team had analyzed the genomes of dozens of indigenous South American groups, focusing on populations in the Amazon basin. What they found in two specific groups, the Surui and the Caritiana of Brazil, was a genetic signal that had no business being there. These two Amazonian populations carried DNA more closely related to indigenous Australians, Papuans, and the Onge people of the Andaman Islands than to any other living human population outside the Americas. This signal, which the researchers named Population Y after the Tupi word for ancestors, could not be explained by recent admixture, by European colonialism, or by any known migration route. It had to have arrived in the Americas at the founding. It had to have been there since the very beginning. If Population Y was in the Amazon, had it also been in Canada? The answer required ancient DNA from Canadian soil, and that is where the story gets even more complicated. The first place scientists looked was to a child. In 1968, a construction crew working near the town of Anzick in central Montana uncovered a remarkable cache of artifacts beneath a rock overhang. Alongside the tools, which were classic Clovis-style points, were the fragmentary remains of a young child, a toddler no older than 2 years old when he died approximately 12,600 years ago. He was the only human burial ever found directly associated with Clovis artifacts. He was, by the standards of the old model, as close to a direct ancestor of all indigenous Americans as science had ever found. In 2014, a team led by geneticist Eske Willerslev at the University of Copenhagen sequenced the child's genome. What they found was more complex than expected. The Anzick child was indeed related to modern indigenous Americans, confirming a genetic link between Clovis people and contemporary native populations. But his genome showed patterns of relatedness that did not fit neatly into a simple single migration model. And notably, he did not carry the Australasian genetic signal in a clear form, which meant either that Population Y was geographically restricted to South America, or that the ancient populations of what is now Canada were more genetically diverse than a single infant's genome could capture. To find the real answer, scientists needed more samples, and Canada had exactly what they needed. Beginning in the early 2000s and accelerating through the following decade, a series of ancient skeletal remains found across British Columbia, Alberta, Ontario, and the Canadian Arctic began to yield usable ancient DNA. Some of these remains had been sitting in museum collections for decades, recovered during construction projects or archaeological digs. Their significance unknown until the technology to read their genomes finally existed. Others were discovered more recently. Each one was a time capsule, a genetic message sent from thousands of years in the past. One of the most significant was a set of remains found in On Your Knees Cave on Prince of Wales Island in southeastern Alaska, just across the border from British Columbia. The individual, a young man who had died approximately 10,300 years ago, showed genomic signatures suggesting he was part of a coastal population that had taken a route into the Americas, not through the ice-free interior corridor, but along the Pacific coastline, navigating by boat through the Kelp Highway that ran from Asia to the Americas. This strongly supported what had been a fringe hypothesis for most of the 20th century, that the first Americans had come partly by sea, not exclusively by land. In 2021, a team of researchers from the University of Copenhagen, Harvard Medical School, and several Canadian universities published a landmark analysis of ancient genomes from British Columbia, drawn from coastal and interior sites ranging from 4,000 to 10,000 years old. What they found was not a single, uniform genetic lineage descending from a common Beringian ancestor. They found multiple distinct genetic populations that had existed in what is now British Columbia at different times, some showing patterns of relatedness that could not be explained by known migration models. One ancient genome from a coastal British Columbia site showed a pattern of allele sharing with Australasian-related populations that echoed the population Y signal found in Amazonian groups. It was not the smoking gun that would settle the debate, but it confirmed that the population Y signal was not geographically isolated to South America. Whatever its origin, it had been present in ancient populations much further north. Even more striking was what the ancient genomes revealed about population replacement in Canada. The populations that had lived in a given region for thousands of years were sometimes replaced, fairly abruptly in genetic terms, by populations with a completely different genetic profile. Not admixed, not blended gradually, replaced. In parts of British Columbia, ancient coastal populations carrying one genetic signature appear to have been largely displaced by interior populations carrying a different signature at some point in the prehistoric past. In the Canadian Arctic, the story is even more dramatic. The people who built the Dorset culture, one of the most extraordinary civilizations ever to exist in the high Arctic, appear genetically to have been a completely separate founding population from the Thule ancestors of modern Inuit people. When the Thule expanded eastward across the Arctic around 1,000 years ago, the Dorset people essentially vanished from the genetic record. Whether through conflict, displacement, or simple failure to interbreed, they left almost no trace in the genomes of any living Arctic population. This finding, which emerged from a 2014 analysis led by Manasa Raghavan and Eska Willerslev, reframed the entire history of the Canadian North. The Dorset were not the ancestors of modern Inuit. They were a separate people with separate origins, and they disappeared. The Arctic was not populated once, but at least twice, by genetically distinct groups with different histories. The implications of this for Canadian national identity are enormous. Canada presents itself as a country built on three founding cultures: French, English, and indigenous. But indigenous is not a monolith, not ethnically, not culturally, and, as ancient DNA is now showing definitively, not genetically. The peoples who inhabited Canada before European contact were not all descended from a single wave of migration. They were the product of thousands of years of complex demographic history, including migrations, replacements, admixtures, and possible connections to populations on the other side of the world. But there is another layer to this story. It concerns a genetic signature that has mystified scientists for longer than population Y, and it generated even more controversy. It is called Haplogroup X. Mitochondrial haplogroups are genetic lineages passed exclusively through the maternal line. The major haplogroups found in modern indigenous peoples of the Americas are A, B, C, D, and X. The first four are clearly related to lineages common in East Asia and Siberia. Haplogroup X is different. Today it appears among indigenous peoples of North America, particularly in the Great Lakes region, and among the Ojibwe, Nuu-chah-nulth, and Sioux nations, at frequencies between 2 and 4%. Outside the Americas, the distinct American form of Haplogroup X is not found in East Asia or Siberia. Instead, it appears at low frequencies in populations across the Middle East, Europe, and Central Asia. For nearly 2 decades after Haplogroup X was first identified in indigenous Americans in the 1990s, this distribution was deeply puzzling. If the founding populations of the Americas had come exclusively from northeastern Siberia, why did their maternal lineages include a marker present in the Middle East, but absent in modern Siberia? Fringe theorists seized on Haplogroup X to argue for pre-Columbian transatlantic contact. Scientists rightly dismissed those interpretations, but they had to acknowledge the anomaly was real. Ancient DNA from Siberia and the Altai region of Russia eventually revealed that Haplogroup X had been present in inner Asian populations during the period the Americas were being settled. It had simply disappeared from Asia in the intervening millennia, replaced by other lineages. The ancestors of the American X2 variant had crossed into the Americas as part of the founding populations, but their Eurasian relatives had not survived to the present in detectable numbers. And this illustrates the same lesson written across every chapter of this story. The founding of Canada was far more genetically complex than any simple model can capture. The populations that existed 15,000 to 20,000 years ago may have been vastly more diverse than anything that survived into the modern era. The ancient DNA record is not just adding detail to the story. It is revealing entire populations that we did not know existed. In 2017, a team of Canadian and international researchers announced the analysis of ancient DNA from a site in the Yukon called Upward Sun River. The remains belong to two infants buried together roughly 11,500 years ago, making them among the oldest human remains ever found in the northwestern part of the continent. The older of the two infants, a female, was given the name Tsachiti Anen Teida Ga'ai by the local First Nation community. Her name translates roughly as Sunrise Girl Child. She belonged to a previously unknown genetic population that the researchers called ancient Beringians. The ancient Beringians were genetically distinct from all other known founding populations of the Americas. They were related to the ancestors of modern indigenous Americans, but they had diverged from that lineage before the expansion that populated the rest of the hemisphere. They represented a remnant of the population that had lived in Beringia during the glacial standstill, and they had persisted in the far northwest of North America long after other populations had dispersed south. This discovery confirmed that Beringia had indeed been a staging ground for the peopling of the Americas, with populations developing there in relative isolation before expanding. But it also showed that not all of Beringia's population had dispersed in the same direction at the same time. Some stayed in the north, becoming a distinct population that left traces in the ancient genetic record, but apparently did not survive to contribute significantly to any modern population. Ancient Beringians, in other words, were another lost people. Another chapter in the human story of Canada that was completely invisible until ancient DNA technology was sophisticated enough to read it. The story of the ancient Beringians also intersects with one of the most politically contentious episodes in the history of ancient DNA research in North America. In 1996, the partial skeleton of a man who had died approximately 9,000 years ago was discovered along the banks of the Columbia River near Kennewick, Washington, just south of the Canadian border. He became known as Kennewick Man. His skull, like those of the ancient Paleo-Americans found in Brazil, did not resemble the skulls of modern indigenous Americans. It bore a closer resemblance to Polynesian or Ainu populations from Japan, and the discovery immediately sparked a legal battle between local indigenous tribes seeking repatriation and scientists seeking to study the remains. When ancient DNA was finally extracted from Kennewick Man in 2015, the results complicated everything. He was genomically most closely related to modern indigenous Americans, and specifically to the Confederated Tribes of the Colville Reservation. His skull had looked Australasian. His genome pointed to Native American ancestry. The lesson this carries for Canada is direct. Skull morphology and genetic ancestry are not always telling the same story. The ancient world of the Americas was complicated in ways that bones alone cannot reveal. Only the DNA can show the true connections, and even the DNA keeps generating new questions. And those connections, as the data continues to accumulate, keep pointing in directions that surprise everyone. Take the question of the timing of the initial settlement. The land bridge model had always assumed that humans entered the Americas sometime after a specific glacial event that would have made the interior corridor passable, with the oldest solid evidence for humans south of the ice sheets placed around 15,000 years ago. But sites keep being found that push that date back. A site in Chile called Monte Verde has evidence of human occupation 14,500 years ago. A site in Mexico called Chiquihuite Cave has yielded stone tools that its researchers date to as far back as 26,000 years ago, though this remains highly controversial. If humans were in the Americas 25,000 years ago, they were there during the height of the last glacial maximum, when crossing Beringia on foot would have been extraordinarily difficult, and the interior corridor would have been completely impassable. This means they either crossed before the glacial maximum or they came by sea. Each of these options has profound implications for who the first Canadians were and where they came from. The coastal route hypothesis, supported by the ancient genome from On Your Knees Cave and by the growing body of evidence for sophisticated seafaring cultures along the Pacific Rim, is now taken seriously by most researchers. The Pacific Rim, at the time the Americas were being settled, was a genetic mixing zone connecting populations from northeastern Asia through island Southeast Asia, and potentially to the margins of Australasia. If the first Canadians came by sea along the Pacific coast, they may have incorporated genetic contributions from a far wider range of source populations than the simple land bridge model ever allowed. This is the context in which the population Y signal needs to be understood. It does not require that ancient Australians literally sailed to Canada. What it requires is that the population from which some of the first Americans descended carried genetic ancestry more closely related to ancient Australasian populations than to any other living group outside the Americas. The founding populations of the Americas were not genetically homogeneous. They included groups with different histories within Asia, some more related to East Asians, and some more related to ancient populations carrying Denisovan, an archaic human ancestry that is disproportionately represented today in Aboriginal Australians and Papuans. When we talk about the first Canadians, we are talking about people who were not simply Proto-East Asians, but a genetically diverse founding population that included some of the oldest and most distinct human lineages on Earth. The story of ancient Canada is, in part, the story of ancient Australia. Not because Australians came to Canada, but because both continents were settled, ultimately from a broader ancestral population in Asia that carried within it an extraordinary range of human genetic diversity. Diversity that has since been largely reduced or erased in Asia itself, but that was carried to the far corners of the Earth by the most daring and long-distance migrations in human history. For Indigenous Canadians, these findings carry a significance that is both empowering and complicated. Empowering because they reveal a history of extraordinary complexity and depth. The ancestors of modern First Nations, Métis, and Inuit peoples were not a single undifferentiated group waiting passively in Asia to cross a land bridge. They were the descendants of multiple distinct lineages, some of them among the oldest in the human family tree, who navigated some of the most extreme environments on Earth, developed sophisticated technologies and social structures, and built civilizations that flourished for tens of thousands of years before any European set foot on the continent. Complicated because the question of who is ancestral to whom becomes harder to answer cleanly when the genetic record shows repeated population replacements, admixture events, and the presence of founding groups that left no modern descendants. The ancient Beringians left almost no trace in modern populations. The Dorset people of the Arctic left almost no trace in modern Inuit genomes. The Paleo-Americans, whose skulls look Australasian, appear in the ancient record but are hard to find in the genetics of any living group. These were real people, real societies, real cultures, and they are gone. Not absorbed. Gone. This is the dimension of ancient DNA research that its most ardent practitioners sometimes underemphasize. The history it reveals is not just a story of connection and continuity, it is also a story of loss, replacement, and disappearance. Canada's past was more diverse and more violent than the textbooks suggest. The populations that existed 10,000 years ago were not the peaceful, unchanging ancestors of the people Europeans met in the 15th and 16th centuries. They were the survivors of their own complex history of migration, competition, and demographic upheaval. Understanding this does not diminish the claims of modern indigenous peoples to their lands or their cultures. If anything, it deepens them. The relationship between indigenous Canadians and this land is not 10,000 years old. It is older than that, rooted in populations that began moving into North America before the ice sheets fully retreated, that adapted to the shifting environments of post-glacial Canada with extraordinary skill, and that built the cultural foundations upon which modern Indigenous nations stand. The fact that the genetic story is more complicated than anyone imagined does not make that relationship less real. It makes it more profound. What ancient DNA is forcing Canada as a country to grapple with is the uncomfortable truth that the narrative of settlement used to justify European colonialism was built on a profound misunderstanding of who was here and how they got here. The idea that the Americas were settled by a single wave of migrants who simply wandered in from Siberia and spread out was always a convenient simplification. The genetic data is showing just how much complexity was erased by that story. The first Canadians were not one people. They were many peoples with deep and tangled roots stretching across the full breadth of the ancient human world. Some of their ancestry connects them to populations in Siberia and East Asia. Some connects them to the ancient populations of the Altai and Central Asia. And some connects them, through mechanisms science is still working to trace, to the ancestors of people living today on the far side of the Pacific. Every year, new ancient genomes are extracted from sites across Canada, from burial mounds in Ontario, to shell middens on the British Columbia coast, to permafrost sites in the Yukon. Every year, the picture grows more detailed and more surprising. The founding of Canada was not a single event. It was a series of waves, pulses, replacements, and encounters spread across tens of thousands of years, involving populations with histories that reach into the deepest chapters of the human story. We thought we knew who the first Canadians were. The DNA is showing us something far older, far more complex, and far more extraordinary. The first Canadians carried within their genomes the ancestry of populations that had lived at the margins of Beringia, on the coasts of ancient Asia, and in regions whose genetic descendants today live as far away as the highlands of Papua New Guinea and the red deserts of Australia. That is the story ancient DNA is telling. Not a story of a simple journey from one place to another, but a story of the full complexity of the human experience. Written in molecules, preserved in bone, and waiting for tools sophisticated enough to finally read it. Canada is not who we thought. And that turns out to be the most Canadian thing imaginable. A country whose true story is more layered, more contested, and more connected to the rest of the world than anyone was willing to admit. The ancient dead are speaking. And what they are saying is that this land, these people, this history belong to a story bigger than any one nation was ever willing to claim.